Okay, so somebody just, maybe Teresa, asked this question to define what a kernel is. Okay, I think I did it yesterday, but I'm going to give a definition again. It's the same. But what is a kernel? So this is a definition of a kernel, and these are meant to be very precise. Kernel K on a finite set X. Okay? If I want to give a formal definition, I did this yesterday, but I did a lot of things yesterday, so I can understand if you don't remember or you didn't understand it. So this is a definition. Kernel on a finite set. Okay, so in some sense, this is not really in the literature, because people have defined reproducing kernels a lot. Reproducing kernel is an object many of you are familiar with. What is a reproducing kernel? And here we're talking about a kernel. Well, this is designed so that people don't have to know things so fancy. I designed this notion of a kernel so people can understand it more directly and don't have to go through the definition and constructions of a reproducing kernel. Okay? But a reproducing kernel, in the standard sense of Ehrenstein from the 1920s or 30s, if you restrict that definition to a finite set X, you get exactly what I'm going to say. But it's simpler. The definition is simpler. Okay, so we understand what I'm trying to do here. Okay, so the definition, it's a function K on the product of X cross X into the real numbers such that, okay, symmetric. Symmetric is very simple, very, very simple. And symmetric, I hardly have to say, k of xy equals k of yx. And this is the key thing, positive, I'm going to put quotes here, positive definite. Okay, now what does positive definite mean for such a function on a finite set? Okay, at first glance, people say, well, maybe K is a matrix, and the matrix is positive definite. Okay, that's about right. But is K a matrix? K is not a matrix. So we just have to, I'm a mathematician, so I like to do things formally correct. K is not a matrix, okay? But now we could take an order on the space X. This is an ordering of these finite set. Any order they exist is just a finite set. No big deal. So we take an order, and once we have an order, we could think of the order, I equals 1. Then we have I plus 1 equals I equals, well, I get mixed up with these things, you see, equals I plus 1. But we just order it and we write the elements. I equals 1, 2, 3.
Too light to see.
Too light.
Too light to see.
Oh my God, what can I do? I can raise this help? Maybe I can push harder. Harder, okay. Anyway, this is, could be interpreted as nonsense, so it's okay if you can't see it. Okay. So anyway, we have an order on X, and you could think of the order giving a function on X cross X in coordinates. The order gives coordinates for K. Okay? We have an order. This gives a matrix representation. So once we have an order on a finite space, then such a kernel has a matrix representation. Okay? And if I try to write it out, I'll probably mess it up. But it's pretty trivial. And once it has a matrix representation, then we know what positive definite is. Okay? So now we have to formally say this is independent of that order, the property of being positive definite. And that's pretty obvious. You just, matrix theory, if you just interchange the rows and columns of a matrix, you don't change property like positive definite. Okay, so that's what a kernel is. That's all a kernel is. It's just a symmetric function on the finite set, which has this notion of positive definite. Nothing more, nothing less. Okay, that answers Teresa's question. Okay, good. And I'll try, remind me from time to time to write harder, okay? So you can see it. Can you read this? Yeah. Okay. Well, it's good you can't read that anyway. Oh, is that okay going up or not? I mean, with the video cameras and so on, or is that causing a lot of problems?
No, it's good.
It's okay. A little better if I can do that. Okay, so let me go over some of the things in a little with more clarity and detail. Remember, we have a set A. This is equal the set of 20 amino acids. And these are amino acids. There's hundreds of amino acids, but there are 20 that our biologists say are basic for life. I say basic for life. Biologists say more or less the same. So they're the things that life are made of. Actually, proteins are what are important for life, but the proteins can be represented by sequences of amino acids. So, and for us, sequences of amino acids of these 20 are central to our approach to all these questions in immunology. I don't say that everything can be reduced to those terms, but in truth, almost everything can. I mean, there is a big question of protein folding, and there's questions of to what extent do these strings of amino acids determine the folding structure, and there are a lot of questions like that. But for the moment and for this talks, I will just assert that so many questions in immunology depend on amino acid chains. So these are just chains or strings of these elements of A. I mean, if A had two elements, you could think of these as sequences of zeros and ones, as computer scientists study all the time. So this is just the objects of computer science, but with an alphabet instead of 2, alphabet of 20. Okay, I want to keep the alphabet of 20. Some computer science-oriented biologists will try to reduce this to an alphabet of 2, but for me, no. Okay. So this is going to be our universe. These chains are strings of these elements of A. Okay. So I want to define a kernel on these strings. So we want to define a kernel. And I'm going to do this more carefully and slowly than last time. K3 hat on finite set of strings of amino acids. All right, how do I do that? And I mentioned yesterday this principle of starting with a similarity on the basic elements of our universe and use that to expand to a similarity on well, in this case, on the whole strings. So I want to make a passage from a similarity on A to a kernel on the, or a similarity or a kernel on A to a kernel on all strings. any finite set of strings. And that's a pretty central part of what we do, is to make that passage. So there is two parts here. One is, what is the initial kernel on A? And last time I just said it, but that is one of the most tricky parts of the whole development. So I want to say K1 kernel on A. And these kernels are very important too, because these kernels contain a lot of, well, they contain a lot of the biology. So we want to get a kernel which holds the biology. So results derived from the kernel will be important for biology. So I guess some people in learning theory, what we're doing is finding a good kernel. So this is a search for a good kernel. Okay, so we start with K1 on the kernel first. Now K1, I said yesterday, Blossom 62, but this hides a lot of the subtleties and problems. I want to now deal with it more exactly. What is K1? Blossom 62 itself is the most important similarity matrix for amino acids in biology. Of the 100 or more mutation matrices, substitution matrices, this is the one more universally used. So I'm going to first indicate what is the usual Blossom 62 and why we don't want it. Okay, so we start with Q. Okay, Q I call the raw data of Blossom 62. It's the raw data, which is very, very hard to find in the literature, because everybody takes this Blossom 62, this 20 by 20 matrix as a starting point. So we want to go back to the derivation, not all the details of the derivation, but some ideas that concern us most. We want to look at the matrix, A matrix Q, which is the raw data for this. Okay, what is that? That's obtained by a process which I won't even try to understand. I never understood it. But the idea is we take all the proteins in the database, I think SCOP, it's the biggest database, contains all understood proteins at a given time. And the idea is one aligns those proteins by a process lining them all up so that the positions correlate. Okay, that's something we never do, but that's what they do for Blossom 62, is take that alignment. Now the alignment, I believe, will introduce errors. So, but we accept that. But, so we accept that what they're doing with these alignments. Now they take, this is the famous people who develop Blossom 62, two names, equal names. What is it? I'm terrible about remembering names. What? What? Hennekoff and Hennekoff. Yeah, right, sure. Hennekoff and Hennekoff. Usually I remember that. But yeah, Hennekoff and Hennekoff do this. And then what they do is they put blocks. They use blocks of these proteins, of these, I don't know how many there were, a few thousand at the time. They put them into blocks which correspond in functions. So they're functional blocks. So in a block, you'll have proteins with the same function. These are all defined using chemical properties or biological properties. And then they look at these alignments, and they do a counting procedure to get a rough kernel. That's going to be what they find is something symmetric, a matrix Q symmetric, positive elements, and they normalize it so that Q is function on A cross A into, now it's into R plus symmetric. They normalize it so it's a probability measure on the product. Okay, so that's what I call the raw data.
Maybe just write down summation Qij equals 1. Not everybody knows the probability.
This is a finite probability measure. I don't like matrices. Okay, you can say, well, if you want to say something, I would say Q of xy, sum over xy equals 1.
Good.
Okay.
That's what I should do.
Okay. Okay. So yeah, it's a probability measure, but these are all positive, so it's just a simple probability measure for finite probability on the product. Okay, now that's been done. We go along with Hennekoff and Hennekoff a bit more. We define a marginal probability. This is a probability measure on A cross A. We define a marginal as follows. P of x, the marginal is going to be a probability measure on the space X. And that's just given by the sum Q of x, y, sum over y belong to X. So there's an underlying uniform measure on X. That makes sense of this. But this gives us a new measure on X. Okay. So we have this. And now what Hennekoff and Hennekoff do is to take Q of X, y, divided by P of X, P of y, for various reasons which I don't 100% understand, but it's okay. We do the same. We call this matrix K of X, Y. It's a kernel. So that's our kernel, just like that. What Hennekoff and Hennekoff do is they go further. See, this is a nice object. This is even positive definite. We proved it. No one else ever worries about these matrices being positive definite. For us, it's everything, because that gives us eventually the distances. If they're positive definite, then we can use that to define distances and many things. So we proved ourselves. We had to. It wasn't any reference, hardly any references to Q in the literature. So we checked the eigenvalues, computed them. They're all positive. So it's positive definite. Okay. And what Hennekoff and Hennekoff do, they don't stop here. They take the log. That's very common in bioinformatics. Take the log. The log just messes everything up. Gives a huge amount of negative quantities. Then I think we surely lose positive definite. And then they round off to the nearest integer. And these are numbers between 0 and 10, something like that. So rounding off to the nearest integer is a gross, too gross. So anyway, that's what Blossom 62 is. So this is going to be our basic K1 right here, except, so here we have amino acids. We want to have a kernel. And so we're not quite finished. And this is the most subtle part which we don't understand. And that is we take beta. So let's take off 1 here for the moment. Then we have a beta which is positive. And that's going to be our parameter. Okay, so this is something that's crucial for everything and something we don't understand very well. This parameter beta. It's hard to understand. Maybe somebody here can give us a good answer. So I want to take K, xy, to the betath power. Okay, now this is not a matrix power. This is a Hadamard power. I don't know how well people know Hadamard powers. I surely didn't before this. Hadamard powers means we take each matrix element, each entry in the matrix, take it to this power. So it's hard to deal with for beta positive. So we take this matrix now, and this is our K1. It depends on beta. So what I've described so far is this kernel that we use on the amino acids. For amino acids, we would take beta to be about 0.08. I think we take beta to be exactly 0.08 for the amino acids. Okay, so this fills in part of the talk yesterday. This says where we start, and it's important because it's one of two contributions to our whole final kernel. All the things we do, peptide binding and classification of diseases, all those things depend. The only biological information that goes into that is in this K1. That's biological information. And in the sequencing of genes or whatever, the sequencing of the sequences, it uses some biological information. Well, it uses quite a bit because the sequences are given usually for alleles by some kind of micro probes, spectroscopic things. In fact, in the World Health Organization, they have a whole list of medical biological methods for determining the classification of diseases. So we do essentially the same classification eventually with only the information that goes into the information in the sequences we use, the sequencing. We use that, and we use this information I just gave you, which has to do with this, the blossom matrix. That's all we use, and we're able to get a huge amount of conclusions from just that about peptide binding problem and about the serotype classifications. Okay, and what's really almost unbelievable is we're able to almost get as good, or maybe it's actually more accurate than the World Health Organization classification. But on all the things, and I'm getting ahead of myself a little bit, on all the genes or alleles that the World Health Organization has assigned serotype group, we get exactly that. Everyone where they've taken the step of making that assignment, they assign the serotype group to maybe a third of the alleles in our big database. And we differ a little bit on some of the ones they didn't assign, give an implicit, it's not really an assignment. On those we differ just a little bit, but on the assigned ones we get exactly the World Health. That's the one they've confirmed by a lot of laboratory reports from labs all over the world. I'm anticipating and giving a motivation for this development of this kernel. So then after that, this is not too much to say. Last time I said something, so we define K2 Riho. K2 of U and V, say sub K. This is if U has length the same as V equals K. Huh?
That was not my question. My question was about if you have more global information in K3
Yeah.
and you want to deform it, can you get the deformation in K1?
I don't know. I have no idea. Yeah, I don't know. No.
So a kind of inverse problem.
Yeah, I know, but I don't have any idea. No idea on that. Okay, so here we have what are called K-mers, chains of length K, chains of amino acids. And then we define K2 as simply the product I going from 1 to K, K1 of UI, BI. This will always be depending on beta, which I suppress. Okay, so this I mentioned last time, and it's fine. It doesn't need any more clarification. Then K3 of two chains, these are two amino acid chains. This is equal to, I better do this a little more slowly. I look at the average over K, U, and V, such that U and V satisfies this. I want to look at the average of K2 of U, V, and I want U to be contained in F, V contained in G. When I say contained, I mean slightly a notation which we haven't maybe arrived at the best notation. This U means a substring of the string F. Substring in the sense that we haven't made any gaps. We just say the same, it's a substring in a strong sense. And we haven't found the right words. It's not obvious. Some people give words that don't make mathematical sense. So anyway, I think people here can understand what I mean when I talk about a substring U of F. And then the final thing, if we have any kernel K on any space, we want to normalize it. So I want to define for K, any kernel, the following: K of x, y, normalized, this hat. This is the correlation normalization of a kernel. And that's equal to, by definition, for any kernel, this is the definition, that's K of x, x, K of x, y, wait a minute. I'm going crazy here. Correlation kernel is going to be, I'll write it up here, it's K of x, y divided by K of xx times K of yy, the square root. That's the normalization or correlation kernel. So it's just a correlation. You can interpret it as a correlation of a couple of vectors. But the idea is the correlation kernel has always got values less than or equal 1. And it's maximal when y equals x, you get 1. And so you can think of it, everything is positive here, so you're in this orthant. And so you can think of this correlation kernel giving you this point in here, which can be interpreted in terms of the projection of this vector on this vector. Okay. Good. And so we want to look at then K3 hat. That's our geometry. Okay, that can be a geometry on, if you want to, on this finite space. I think of this as, in fact, a geometry on the finite space X, where X is some set of amino acid chains. And there's one thing I left out here, which is important. Maybe I should say it. Is this is a kernel. All these are kernels. Okay? So these are kernels, and it doesn't look like much, but it is a lot. And it's not obvious that they're kernels. Okay, so they're kernels means they're positive definite. Okay, so if K1, if this is positive definite, then by general nonsense, K2 will be positive definite. That's easy. And if K2 is positive definite, then K3 is positive definite. That's easy. So the question is K1 positive definite? That's not easy. Because this Hadamard power beta is not just matrix multiplication. So there's some subtlety involved in showing that this matrix, I call it K1 beta here, is positive definite. So we have a theorem. This matrix is positive definite. Or this kernel is positive definite. Okay, so how does that go? Well, we found a linear algebra with Horn, H-O-R-N, is one of the authors. And in that book, they have a theorem which says this is going to be positive definite for all positive beta if the Hadamard log is conditionally positive definite, if and only if. And so conditionally positive definite means that it's going to be positive definite on the set of matrix of the elements V such that V sum of VI equals 0. So that's going to define a linear subspace of V. And so we had eventually to check that this log of K without the beta satisfied this positive definite on this subset, conditionally positive definite. We checked it. And so from that we conclude this theorem. By using the theorem one can find in Horn, which is not so trivial, that a condition for Hadamard power to be positive definite for all positive beta. They have this necessary sufficient condition, and that's good enough for us. So that's the starting point of positive definiteness, and so that eventually gives us a positive definite kernel here. So that means we can operate with that geometry on the space. Okay, so we want to look now at the peptide binding problem. This is a first big open problem in immunology. Peptides are fragments of proteins, definition, but they are usually fragments of what one could call bad proteins. Viruses, proteins associated to bacteria or viruses, and such things as that. The fragments of those. So the traditional idea of vaccines going back to Jenner is if you take some of these fragments or part of a diseased animal and use it for a injection, a human injection, it's going to prevent that disease. So that's the starting of the history of modern immunology, using these peptides to create immunity. So the key step in that process is there are a binding of a peptide to an appropriate cell in the human body we call MHC2 cells. Not cells, or proteins, or molecules. These are molecules in each cell of our body, and they are especially designed to deal with disease, so they're associated to our immune system. And they are proteins which are derived from genes. So there's a gene process. There's about 10 dozen genes that we, in our process MHC2. And each of these genes has a lot of variance, a lot of variation going over the population. So a gene might have a representation in one person, a different representation in another person. And those differences make those people susceptible or not to certain diseases. It gives a disease profile for a person. It's a human genome. And also, almost as important, for organ transplants. If you have an organ transplant from somebody with a little different genetic complex representation of these genes, then the body will reject it because they think it's an invader. And so that's why in immunology is a huge amount shown to when are two alleles similar. Will they have the same binding properties and the same properties when it comes to rejection of a transplanted organ? So that's, in a nutshell, just where this is going. Okay, so the first thing is this problem of peptide binding. So peptide binding, let me describe peptide binding. So the peptides you could think of as these fragments of dangerous viruses or bacteria. And they're small, they're fragments, so the fragments range, well, in this class that we're working in, MHC2, they're averaging about 15 length of their amino acid chain. The fragments of proteins, and proteins all are represented by amino acid chains, and the peptides are fragments approximately of length 15. They could be in one class more like 9 length, or it could go up to 25 or 30. But those are the peptides, and those are the things that we want to know if we're going to prevent diseases. And so we have to satisfy this main step of binding to an appropriate immune cell, and the surface immune allele, which can be represented in the surface of a cell. And when that binding takes place, very often, I'm simplifying things a bit, that bound protein alerts human cells who see it to fight and to kill that cell, killer T cells. So that's why the peptide binding is so important, because it's the main step in this immunological reaction, which sets off this process of killing the infected cells by these T cells, helper cells, B cells, and so on. Okay, so that's a little bit of the immunological background. So these peptides are amino acid chains. Okay? And now what we want to do is look at the fixed allele problem. So I want to fix an allele. Allele is now the representation of a gene, and we're talking about genes associated to immunology, immunological system. Allele is a representation of a gene in a person. So we say a gene is polymorphic if it will have maybe 100 representations in different people. And anyway, so these genes have these representations, and the alleles that we deal with, the word here, which I won't try to even say, MHC, we work with 2. It's called, I can just barely beginning to learn this word, major histocompatibility complex. And I believe that the methods for MHC1 do not go over to this in general. Okay, and so what we have is then associated to these immunological alleles, we have a set of peptides constructed by doctors and medical research. I'll write P sub A is a set of peptides. And so this is set from 100 to 5,000 peptides. And these are candidates for binding. Peptides in this set might or might not bind to this allele. So binding of a peptide, and let's say the set here, to an allele is this crucial step we really want to understand. That's a basic step in creating new vaccines. It's simplified a little bit here because I fixed the allele. Okay? And so what I want to do is to look at P sub A. Actually, what I've written here is P sub A, the set of peptides with medical data. So there is a database of X belonging to PA, Y belonging to the interval 0, 1, so that for these pairs, X, Y, the medical practitioners have given us a binding number. Y is a number between 0 and the binding of X to Y is given relatively between 0 and 1. So here we differ from almost all other people working in this medical field. We center on the idea of peptide binding should be a real number, because binding is a matter of degree in medicine. I mean, here we have a molecule A, here we have a peptide P, and so it could bind like this, or it could bind a little farther out, or it could bind like this. So binding is a matter of degree, the degree being given between 0 and 1. And we would like to know that in general, and we have data already on P sub A, so we can take a larger class of peptides P and use the data given this way. You could say XI, or XI is a peptide in this database. YI belongs to PA cross the interval 0, 1. Okay, so the data is of this form. Okay, so there are a few of these A's with a lot of data. Not many. I forget, 14 maybe, with a really good data. We'll see them in a few seconds. Okay, so now what we want to do is to find peptide binding functions. Say F sub A going from P to R. This is mainly just the interval 0, 1. But when you find it, unless you project it onto here, you get something a little bigger. I could put 0, 1 here and I wouldn't lose much. So I want to find this function, F sub A. That's our problem. Okay, so to do that, we use supervised learning theory. And so one uses least squares here. Regularized least squares, supervised learning theory. So we look at the problem, minimize F. I can say over what in a moment. So F of xi minus yi squared, sum over I, plus lambda is a penalty. This is common in this kind of mathematics. Then we write norm of F k squared, k hat 3. Okay, so here's where we use our kernel. The kernel plays the role of well, it's an inner product on the Euclidean space, and these functions F are also other functions on the peptides. So they're in that space. So I could say F belongs to H. And here we could write even P bigger than the data space. So P is a set of peptides which is bigger than the data space. Okay, so we want to look at this problem. And k hat 3 is exactly what we've been developing all along, that carries accurate biological information, and we have to see if it's really going to work. Okay, so now let me do this. If I can do this. Okay, so here's our results. Solving this problem, it's a little bit of a computational problem. I mean, there's a lot of learning theory. I spent many, many years of my life studying the theory of this problem, not quite so much the computation. And there are these methods for solving this. It's linear algebra, because this is quadratic. So this is a linear algebra problem. For each lambda, one has to choose lambda computationally, and one does it by something called leave one out. For example, we did. And there's lots of ways to check the prediction. But solving this will give us this FA. Okay, so here are the results we got. So these are the alleles. These are the way that the alleles are represented in this great database by the World Health Organization. And this notation changed every few years. So there's a lot of problems with everything being updated. The whole structures are updated. But this is pretty reasonable. I think this was the maybe 2008 picture, and that is held with a lot of people. People get tired of changing the notation too often. And let me just say what that means. Before the star here is a gene. These are genes. So here we see, a dozen genes or so. Yeah, so those are genes, and they're usually different. These are just DRB star 1. This is all the same gene. Then we have 3 more. So there's 4 genes here. Okay, the next element here, after the star, is the family. So here we only have one family listed, and here we have DRB star 1. Here, almost a dozen families are listed right down here. So these are families. Hundreds of times are tables of about 800 of these. There are 800 of these that are in this class MHC2. And I study this a huge amount for all kinds of things. And the tables by this WHO organizations, they'll give you a lot of properties of each one of these alleles. These are alleles, and so the family, as I say, the second two after the star, and the next two give you the number in the family. And these are the alleles with the most data. So many papers written on peptide binding problem for these alleles. These are kind of a testing set for what you're doing. So there's a vast literature, a very big literature on that. And the best work, which we aspire to, is this Nielsen, Martin Nielsen of Buenos Aires now. He is a person who has had the best numbers here for accuracy. This AUC is area under the curve. It gives you the accuracy of the prediction. And Martin Nielsen, he says in the paper, they say in their papers, and they're right, that this is the state of the arts. It beats every other of the algorithms and work for computing the peptide binding functions. And so here we're very happy because we have a little better. Okay, yeah, so you can see this. This is the accuracy, and this is the number of peptides in the data set, P sub A. If A is an allele in this column, the number of peptides is this. So you get quite a lot of picture just from this here. And the AUC is something, area under the curve. It's a measure of accuracy. And these are more, these are less common, these 3, 4, 5 star. But these, as I say, have been discussed in terms of these families or serotypes. And that's my main source of my lecture tomorrow, are classifying alleles into families which correspond to the World Health Organization serotypes. Okay, let me give you the next table. Here is the maybe more important. So this is what we call panallele. So we take 24 alleles, and then we take data from some of them. So we have a data, I think the data is about 33,000 points. There are going to be pairs of peptides and alleles. So we get a big data set in the product space. They're in the product space, peptide space, allele space. So here we're going to be using the same kernel on the allele space. On the allele space, you have about 800 alleles. Their length 80 or so, about 80. And we use the same k-hat3 with a little different beta. We make beta a little bigger because they're longer. So we make beta equals 0.12. And then we do the same process, except now we work with pairs in the product space. So we want to do the same least squares. So we want to have a kernel on the product space, on the space of peptides product alleles, because that's where we're getting our new peptide binding function. It's going to tell us, given any, eventually any allele combined with any peptide, what's the chance, how likely, how well does it bind? So this is a very broad program. And so here we say we take beta set to be 0.1 for the allele kernel, 0.12 for the peptide kernel. And then we you do the same least squares here. Same thing. Appropriate choice of lambda and so on. And here we get these numbers. Again, Nielsen and the people around Nielsen have the best, they have the state of the arts on this. Okay, so we're always comparing what we do with Nielsen. And here, how do we do? On this problem, Nielsen has accuracy better than ours. On the weighted average, we do a little better. very little better. Okay. Anyway, so this is what we did. Well, it's a long story, but let me just talk about that today and then go on to another couple slides. Okay, here's a new data set. The first one was done with a Nielsen data set of 33,000 pairs. And then we started with a complete new data sets just to see how get independent verification of what we're doing. And then we have weighted average. We lose a bit to Nielsen on this. And the average we lose significant. But, as I said, we believe that thinking of peptide binding as a 0-1 problem, where you have binding versus non-binding, it's not such a good criterion because you want to know the degree of binding. How much is the binding? What's that number between 0 and 1? And so in that case, what we could do is using Nielsen's algorithm, make this comparison. Let's see if I have it here. Oh yeah, so here we get RMSC. That's a accuracy estimate for the real valued function. And here are the numbers for that. And with RMSC, smaller is better. So here we do better than Nielsen across the board for this concept of accuracy. So anyway, that's where our work stands on these problems of peptide binding. These are the main big problems of peptide binding. The pan allele, which is more recent and I think more satisfying, and the fixed allele, which came earlier and has a much bigger history. And this is what we got. Well, I think I kept you for a long time. Thanks very much.